So good morning everyone. Today I'm going to share the topic performed by Haluka. Tentative title is Stephanie's tune macrophage polarization in viscoelastic matrix. So I'm going to share certain background for starting the presentation. So as you know, we already knew that 2D stiffness increase macrophage polarization by biochemical cues. So when they regrouped 2020, they found that stiffness increased YAP, localization, as well as TNF-alpha, interleukin 10, amyloid-related cytokine increasing over stiffness. As you know, they utilized PAH from 1 to 200 kilopascal. After 24-hour sitting, they treat 6 hours LPS. And then Dong-Hee Kim group, they did similar experiment for M2 polarization. So from 0.2 kilopascal to 35 kilopascal, they also find out under biochemical M2 cytokines, interleukin 4 and 13, transcription factor STAT6, M2 STAT6, they are increasing over stiffness, while M2 related cytokine gene, LET, NLA, this and RG1 are also increasing under interleukin 4 and 13 biochemical factor. They also utilize PAH through this range of kilopascal. After 24-hour sitting, they treat 2 hours for interleukin 4 and 13. Then from left and right, M1 or M2 polarization, we can find 2D stiffness increase macrophage polarization into M1 and M2 respectively under certain biochemical cue. But without biochemical cue, like low stimulation, we cannot see any interleukin TNF-alpha secretion and interleukin 10 secretion, no change. And in terms of M2 related cytokine or gene expression level, also no change, almost. So only biochemical factor, biochemical cue can drive the macrophage M1 or M2 polarization on the stiffness. Recently, the David Murray group, they culture the monocyte in 3D gel. So they found out 3D stiffness or stress relaxation increase monocyte differentiation to dendritic cell. Here, in summary, they utilized collagen arginine 3D gel from 0.7 to 7.5 kilopascal. And then how they did the experiment? 1 hour gelation in 3D, and then 1 hour accumulation in HBSS, and then 3 days biochemical induction using this GM-CSF, interleukin 4, and PGE2. Those are dendritic cell differentiation biochemical factors. So they already target the dendritic cell. So that's why they are using these 3 combination for differentiating this monocyte to dendritic cell. Under this condition, let's see. So this is their schematic images, how the monocyte behave in 3D gel. And then, as you know, this is the viscoelasticity. Over time, their initial stress are relaxed, decreasing. And then when we think about the elastic means over time, the stress is maintained. So we can say this is a stress relaxing gel or viscoelastic property. This elastic property. So here they made 7.5 kilopascal viscose and elastic gel, similar stiffness, while they have 10-fold change of stress relaxation time. For example, elastic gel around 10,000 seconds, while viscoelastic gel below 1,000 seconds. So almost 10-fold change. And then from lost angle tangent, also they can see the difference of viscoelasticity between viscose gel and elastic gel. And then they utilize 4 different groups, stiff and soft. Soft is 0.7 kilopascal, stiff is 7.5 kilopascal, and then viscose elastic. elastic is like this, stress relaxation time is 10,000 seconds, while viscous below 10,000 seconds. So they found that significant difference in elastic stiff gel compared to others. Here, they are utilizing two different donors, donor 1 and donor 2 monocyte. From these two different donors, they found out stiff elastic transcription level are significantly different from others. Here, this soft viscoelastic and then stiff viscous are gathering each other very closely, while this stiff elastic monocyte culture in 3D gel, they are far from others. So they highlight this stiff elastic, and then in the end, they made the conclusion: stiff elastic versus stiff viscous to have one parameter. They do not want to change two parameters altogether, so they focus on stiff and then how viscoelasticity or elasticity can tune the monocyte differentiation to dendritic cell.
저기 보면 비스코스한 패널에 오른쪽 보시고 저기 컬러 있는데
이거요?
비스코스한 데서는 스틱하고 소프트 차이가 별로 없지.
여기서는 그랬어요. 여기는 바이오케미컬 쳤기 때문에 under biochemical factor. But I will show you later. So here they highlight this point, but when I look at the other panel in supplementary, so stiffness seems to affect the monocyte differentiation due to dendritic cell in viscoelastic gel. So I want to ask, stiffness only can make the difference in viscoelastic gel. So I believe this 3D viscoelastic gel and then two different stiffness can make the microbiome environment more biomimetic condition. Here, when you look at this NF-kappa B's gene expression level, even though they didn't highlight that much, but so I want to highlight viscous gel, soft and stiff. Is there any difference or not? Here, okay, see some tendency. And then LB, this is two different donor, same donor, same donor. Also, we can see the donor-dependent manner, but here we found out this donor 1, we can see some trend, stiff, soft, some change. MMP9, this donor, second donor, also we can see some change. So, okay, we can get some conclusion that under this biochemical factor, even viscoelastic condition, soft and stiff, can make some difference. And here, in supplementary data, we can also find out they tried 0.5 kilopascal and 3 kilopascal in viscoelastic gel. Here, they put this data in supplementary, but we can see that 3 kilopascal and 0.5 kilopascal could see very significant difference of gene expression level. Also, we can see STAT3. As you know, STAT3 is M2-related polarization and transcription factor. They are increasing in 3 kilopascal under this biochemical factor. So they didn't highlight that much, but I'm pretty sure that they will do some work about this. Viscosity gel, what is the meaning of stiffness? And then also here, they found out F-actin in elastic gel is independent of collagen adhesive ligand and ectomycin contractility. Here, they didn't add any collagen, and also they found out F-actin is well developed in elastic gel. And then when they treat blebistatin, myosin ectomycin contractility inhibitor, F-actin is probably maintained. So actually, and then when you look at the pan-actin viscoelastic, so even though we can see some difference, but no collagen, which means there's no cell binding moiety, and then when they lose the ectomycin contractility, F-actin is no change. So this is a way abnormal situation, I think, because because here they are using elastic gel in 3D performance. So That is why we already know that F-actin is a very, very promising cytoskeleton component to regulate the monocyte or macrophage differentiation or their polarization. But here, even we remove the collagen in the gel, or if we remove ectomatic contractility, F-actin will change. So I think this is way extraordinary situation, what I believe, compared to what I believe. So here, so previously I mentioned I can see little difference between soft and stiff, even in viscoelastic gel. And why viscoelastic gel is important? So already, as you know that, Charles already mentioned many tissue, adipose, liver, brain, hematoma, or bone marrow, they are all stress-lasting. This is a basic concept of the tissue. Stress should be relaxed by remodeling of the ECM. And then when they look at the data, like bone marrow, breast cancer, adipose, brain, liver, embryonic tissue, muscle, hematoma, skin, all the cells lasting time is below 10,000 seconds, 1,000 seconds, right? So from 100, 1,000 seconds, they are all stress relaxing. But previously, what is the viscoelastic? Below 1,000 seconds, and then elastic is above 10,000 seconds. So I believe if we want to mimic the real condition of the tissue, we have to utilize the viscoelastic gel and then should compare soft and stiff. So this is my understanding. So 2D stiffness elastic gel on 2D, M1, M2 already done. And then 2D viscoelastic versus elastic, 3D, this one, actually this part, 3D and the viscoelastic versus elastic, David Muir group already did under biochemical factor and from monocyte to dendritic cell. But I want to ask, in viscoelastic gel condition, 2D stiffness and the 3D gel stiffness, how this stiffness can make the difference of macrophage polarization. This is our study. So I believe this study can more mimic the tissue microenvironment. So from now on, we can show the result. So first, we are asking 3D stiffness in viscoelastic gel tune the macrophage polarization without biochemical factor. This is our question. So for answering that, we are making 3 different gel. Same component, collagen arginate, but we can change the molecular weight and calcium concentration to make the 3 different stiffness. And then we can see very similar morphology. Pore size is similar, a little similar, and then while pore size similar, and then swelling ratio, dry weight, and then protein diffusion are similar among 3 groups, but elastic modulus is changed.
When stiffness increase, I think both sizes decrease, no? Because when we have more higher stiffness, so both sizes decrease.
Yeah, they can probably decrease.
Arginate test, no?
If we also pneumonia group and GP group also study, depending on the blood outcome also may be caused the different sizes, and that's why we can calculate for the
Okay, so anyhow, we are making these 3 different gel. Initial elastic modulus 1, 5, 10 kilopascal, but stress elasticity is below 10,000 seconds, 1,000 seconds. They are almost around 300 seconds. And then we cannot see any toxicity from this gel in 3D encapsulation. Okay, live and dead, and the K67 positive cell almost similar. Okay, we can make 3 different gel, viscoelastic gel, and then 3 different stiffness. So what is the effect of this gel for macrophage polarization? We can select THPN cell, human monocyte, and we are differentiating them to macrophage by PMA. So we select 1 and 10 kilopascal, and then check INOS, M1 marker. We can see very dramatically decreasing the nucleus volume, while INOS increase. The P65, NF-kappa B, transcription factor, protein, they are empty in 1 kilopascal, while 10 kilopascal highly localized. This is after 24 hours. So we already did same manner, 1 hour dilation, 1 hour incubation, and then 24-hour culture, only PMA. So this is the quantification of nuclear translocation as well as ionization level. We also checked by the qPCR for 3 different time points, 4, 12, 24 hour, 1 and 10 kilopascal. So continuously 10 kilopascal can show TNF alpha increase, and then others, most of them increase. But depending on this M1-related cytokine, someone is early M1, someone is late M1, but we can see some trend. All of them show increase of this cytokine, M1 cytokine expression level increase in 10 kilopascal. And then we also find out from the gene expression level, taline and FAK is highly upregulated in 10 kilopascal. But this is a really big opposite from our ICC result. Anyhow, by Western blot, the P-STAT1 is highly polarized, phosphorylated in 10 kilopascal. And then, while they are phosphorylated, K9 methylation 3 heterochromatic marker, they are also increasing. So we can feel that THP1 cell in 10 kilopascal, they show M1 polarization. And then, while the M1 polarization, DNA nucleus is shrunk, and then, along with K9 methylation 3 increasing. And then we also check the BMDN, bone marrow, mouse, macrophage. Also, if we 3D culture this cell in 4 different gel, we can see stiffness-dependent ionization increasing by Western blot and then other gene expression level. So we can believe that this is not only from THP cell, but also BMDN can show similar tendency. Stiffness can increase polarization. Then we look at the cytosol change in detail. So 4 hours, we can see little like cytoskeleton affecting their try to spread. But 10 kilopascal already 10 kilopascal macrophage, they are losing their affecting expression. So this is a very dramatic change over the stiffness increase, only 4 hours. And then when you look at this cell after 24 hours, we can see many spreading cells in 1 kilopascal, while 10 kilopascal more shrinked. So cell volume and interactive 3D surface area and blabbing cell. Blabbing cell means how this macrophage have this blabbing structure. And then effect intensity, quite stiffness dependent manually. We can see the trend. And then we can also see the affecting and ioness negative correlation and ioness and cell volume negative correlation. So 1 kilopascal, they can maintain the cytosol while they are remodeling the ECM. And then cell volume maintain, affecting intensity maintain. But 10 kilopascal, they are gone. So when you look at the PMS level by ICC, so near the nucleus, PMS level look decreasing in 10 kilopascal, while 1 and 5, they are maintained. And then 24 hours later, this image also show the tendency between nucleus volume and cell volume. And then 20 kilopascal, they are losing affecting, and then they are shrinked. So we also see this similar tendency.
So any reason why means actin is decreased and higher the stiffness?
Yeah, so I will later show you, but the answer is we believe that. So, how can I say, when macrophage cannot remodel the ECM properly, they secrete inflammatory cytokine by themselves, and then they uptake it again. So this kind of vicious cycle can inflame the macrophage into M1 phase. So because we already know that when macrophage is treated by LPS, they are shrinked. I mean, especially nucleus is shrinked.
One my suggestion maybe this is also possible because when you increase the stiffness 1 so number of the photosome
Mm.
start to decrease because when microphage cell is going in M1 polarization, so number of the photosome decrease, yeah, correct, yeah. Number of the photosome decrease, this is the reason also possibility to actin, maybe.
Yeah, it might be true.
Yeah, this is also possible, yeah.
Yeah, actually from the 3D analysis Haluka performed, but we cannot see very clear photosome in 3D structure. Yeah, anyhow, we try to track that photosome structure.
24 hours you can check possible there.
Pardon? 24 hours, yes.
Yeah, 24
Mm.
hours.
Okay.
Because I also, in my fiber case also, I feel this kind of things.
Mm. Okay. We will look at the photosome structure as well. So anyhow, we want to do this ECM remodeling allowance experiment. We already did. Haluka did this experiment for another project. So we already set up how ECM can be remodeled in 2D condition. So we want to apply the same concept in 3D using bead. So we believe that 1 kilopascal soft gel, they allow more ECM remodeling, while 10 kilopascal, they are restrict. Cell cannot remodel the ECM that much. After that proving, so we also look at the nucleus change in detail. Here, 1 and 10, 24 hour, very condensed in 10 kilopascal. Okay, and then nucleus 3D structure, look, they are more flattened in 10 kilopascal because they are shrinked. And then in 3D, volume decreasing. And it is a very interesting nuclear volume time attack. So when you look at the 1 kilopascal, except this 1 hour, they are relatively maintained. While 10 kilopascal, initially they are similar, but they start to decrease. So we saw this tendency, the nucleus volume, cytogen volume, they are not increasing or decreasing. They are not increasing in 1 kilopascal, they are maintained. While 10 kilopascal, they are decreasing the nuclear volume. So we believe this is shrinked. So laminase level is increased in 10 kilopascal. As you remember, H3 kinematin 3 are increasing in 10 kilopascal. So laminase is also increasing. So we believe this is kind of apoptotic or amyloid-related signaling that can enhance the laminase and kinematin 3 increasing. So this is their finding. So from this 3D laminase level and lamin B1, laminase, both of them, they are quite highly expressed in 10 kilopascal. So as you know, we learned from the previous our presentation, Sun 1 and 2, LPS, when LPS treated, Sun 1 or Sun 2, they are decreasing. So let's visit this result published in Nature.com. So they treat the macrophage, PM, and on TCP and treat the LPS. Over time, Sun 1 and 2 are decreasing because here they found out activate macrophage. These Sun 1, 2 are removed from nucleus, and then they can develop more intermediate filament and other cytoskeleton. And this is the phenotype of activate macrophage. So we want to check in our condition how Sun 1 and 2 are expressed. They are also decreasing or increasing in 3D condition. And then next discussion point is that we already learned that 2D condition, LPS treatment, decreasing laminase level. The Vogel group, in iScience paper, they already found that this LPS treatment, that can remove the laminase in peripheral area. Like that here. Also they found out over time on the LPS, laminase is decreasing. They found out in very different experiment. But in our condition, laminase is rather increasing. So we believe 2D, 3D, this dimensionality can make the difference. And then we didn't actually add any like biochemical factor for M1 and M2. So maybe this is a different point between this condition and our laminase increase. And then chromatin change. So we checked H-3. H-3 is increasing. This makes sense because we already know that H-3 and F-actin are connecting each other in cytosol. But when F-actin, they are gone. H-3 can easily localize in nucleus. So along with our expectation, H-3 is highly detected in 10 kilopascal. We believe this is F-actin dependent manner. And then other oscillation, 1 kilopascal enhanced, and canine maintenance 3 decrease. Not only canine methylation, 27 methylation, they are decreasing here. Increasing here. So from the chromatin change, we can say that 1 kilopascal they maintain euchromatin structure, while 10 kilopascal they come to be heterochromatin structure. And our remaining question is that which parameter drive initial inflammation? So say ECM remodeling, number 1. Number 2, cytoskeleton volume or nuclear volume, or cytoskeleton component, or autocrine effect, or chromatin remodeling. So this we have to make the order. Which one is the one first? Which one is the last? So for answering the question, we are setting up the plan for using this inhibitor or activator, low activator, BLAB, osmolality change, Lade, NOCO, Vidaferon, and interleukin 1 beta TNF alpha secretion level, H-stack inhibitor and methylation transferase inhibitor. From this multiple cytokine, multiple activator or inhibitor, say, we can answer which one is the first, which one is the last. But from our understanding, we hypothesize like this. First, ECL remodeling allowance is different between soft and stiff in 3D gel. And then, maybe chromatin remodeling can be performed by physical cue. And then, because of this chromatin remodeling, small amount on interleukin 1 beta or TNF alpha, they are secreted. But they can be auto-uptaken by the macrophage. And then, this interleukin biochemical factor also accelerate chromatin remodeling by biochemical cue. And then, finally, cytosol volume decrease by autocrine inflammation, and then nucleus volume also going down. So here, previously, Grace presented this paper, LPS, on 2D condition, decrease the nucleus area and volume. We also know that the nuclear volume decreasing is a marker of inflammation on 2D. So might be, or if we treat 3D gel, soft, and then treat the LPS, and then this similar chromatic condensation happen in 3D viscoelastic soft gel, and then we can say that this whole phenomenon is from the autocrine effect of macrophage by biochemical cue. So, and then as a final clinical implication is we can link this phenomenon in OSCC, where we already performed subcutaneous gel implantation in the mouse, and maybe keloid or fibroblast or brain tumor or glioblastoma. After finding the key finding in vitro study, we try to link this in vitro to clinical implication. Okay, thank you.